Hello and welcome back. It's Selma with another review of the effects of lead on vertebrates. Lead is a heavy metal that has been known to be dangerous for a very long time, since the Romans used it to sweeten their wine and craft intricate plumbing systems from the toxic metal. Some historians believe the extreme and widespread level of lead poisoning in the Roman Empire even played a role in the empire's eventual downfall. Lead continued to be used for hundreds of years afterwards in plants, pipes, jewelry, cutlery, and eventually, gasoline. In 1921, in an effort to solve the problem of engine knocking in cars, Thomas Midgley Jr. invented tetraethyl lead, branded as just ethyl. Ethyl would go on to become one of the most used gasoline types in the world and caused widespread pollution and lead poisoning that people continue to suffer from in affected areas today. Ethyl was banned in 1996, which is why you can find unleaded gas labels at old gas stations today. Veritasium has a wonderful video on the history of tetraethyl lead and everything that came of it. It will be linked down below, and I highly recommend you watch it after watching this video. All other sources used to make this video will be linked below as well. Today, thousands of researchers have devoted their studies to understanding how lead affects the body and causes the medical and environmental harm that it does. The Mayo Clinic lists the known symptoms of lead poisoning, including developmental delays, seizures, and weight loss in children, and high blood pressure, mood disorders, and infertility in adults. A fascinating study shown here, conducted by Lee et al. At the Harbin Institute of Technology in 2022, sheds light on what may cause some of these symptoms on a molecular level. The study, titled Lead Exposure Causes Spinal Curvature During Embryonic Development in Zebrafish, uses zebrafish as a model organism for vertebrates. The team introduced lead to embryonic zebrafish, which caused symptoms such as delayed hatching, increased heart rate, and malformations, namely spinal curvature. Their research shows that the cause for spinal curvature was found to be endoplasmic reticulum stress and apoptosis. They found that calcium was a good rescue method against lead as well. Let me explain why calcium is significant. The endoplasmic reticulum synthesizes proteins, metabolizes lipids, and stores calcium, to name a few of its functions. Lead is similar in size and structure to calcium and interferes with systems that use or rely on calcium in the body. Lead interference with the calcium in the endoplasmic reticulum can cause stress and lead to apoptosis in cells, also known as cell-directed suicide. Introducing an excess of calcium can help reduce stress and reduce apoptosis in cells, therefore reducing rates of malformation in the zebrafish. Let's define some terms used throughout this study before we get into it. The abbreviation HPF means hours post-fertilization, indicating how long something is introduced or observed in the zebrafish eggs after the eggs have been fertilized by sperm. Pb2 plus indicates the lead ions that accumulate and cause toxicity in vertebrates. Ca2 plus indicates calcium ions introduced as a rescue method. Calcine is a yellow-green fluorescent stain that binds to calcium, which can be used to view any tissues containing calcium, namely bones and muscles. Researchers used this stain, which has little adverse effects on zebrafish, to view the level of skeletal calcification, ossification, and somite development, as we'll see later in this video. I just threw a lot of science words at you, so let's break it down. Skeletal calcification is the process of calcium crystallizing to form bones and teeth. Skeletal ossification is another process of bone formation where cells named osteoblasts lay down new material to form bones. Somites are structures in embryos that are the starting point for vertebral column and skeletal muscle formation. Monitoring these developments is crucial in this study for the formation of spinal malformations in zebrafish embryos caused by lead exposure. Now that we know some of the basic terms used in the study, let's take a look at what regular zebrafish development looks like. As you can see, they begin forming at 4 HPF, begin growing a tail at 24 HPF, hatch between 48 and 72 HPF, and continue growing into a normal fish from there. Now that we know the basics, let's dive into the study, pun intended. To conduct this experiment, fish were split into groups and exposed to 0, 0.1, 0.2, 0.4, 0.8, and 1.6 milligrams per liter concentrations of Pb2 plus ion solution, with the 0 group used as the control. The groups were then tested for exercise frequency, heart rate, hatching rate, malformation rate, and survival rate. For comparison, according to the CDC, human children are diagnosed with acute lead toxicity at 0.7 milligrams per liter concentration of lead in their blood. Exercise rate was measured as an indication of issues in the central nervous system. As you can see in Figure A, every group above 0.1 showed significant decrease in exercise frequency, indicating damage to the nervous system at the low and high concentrations of lead. Heart rate in Figure B was increased at concentrations of lead from 0.1 to 0.8, with the most significant increase from 0.1 and 0.2 This indicates that lead stimulates the circulatory system at an early stage of development when introduced early. Hatching rate in figure C decreased as concentration of lead increased, indicating developmental delays as a result of high exposure to lead in zebrafish. The survival rate in figure D showed that zebrafish at low concentrations of lead were more likely to survive at an 80% survival rate, while the 1.6 group showed only a 20% survival rate at 120 HPF. The malformations measured are truly the most interesting part of this study. At 120 hours post-fertilization, there was a 100% malformation rate in every group except for the control, with 5 common malformations identified. The malformations seen most often were curvature of the spine and a lack of the swim bladder. Sometimes they were even comorbid. Pericardial cavity edema, swelling of the heart lining, increased as concentration of lead increased. The 1.6 group showed a lot of brain hemorrhaging, and the 0.4 group showed an absence of pigment. Based on all the information we just went through, the researchers chose to focus on the 0.4 group for the rest of the study in locating the mechanism of spinal curvature. This was based on their survival rates and malformations, which were significant but not as deadly as the higher concentrations, allowing the spinal curvature to be studied. In this group, spinal curvature began at 72 HPF, showing upward curvature in 50% of fish. At 96 HPF, 85% of fish showed the S-shaped curvature, and at 120 HPF, 100% of fish in the experiment showed downward curvature, as you can see in figures A, B, and C respectively. A calcium rescue group was introduced, where 2 millimole per liter of calcium was used as a rescue method for the 0.4 group. This group showed a similar malformation rate to the control group, meaning that calcium was successfully able to counter the effects of lead on spinal curvature. The control group, the 0.4 group, and the 0.4 group with calcium all underwent calcine staining at 120 HPF to monitor ossification and somite formation. Ossification and somite formation in the lead-exposed group was smaller than both the control group and the calcium rescue group. The lead-exposed group also showed uneven sarcomere growth, where some were short and some were long. To further understand the cause behind lead causing less ossification, less somite formation, and shorter, uneven sarcomere growth, researchers sought to understand the mechanism on a cellular and genetic level. The researchers found 5 genes that were significantly downregulated upon exposure to lead, causing poor ossification and somite formation. They explained the role of each, stating, LECT1 is expressed in cartilage and skull structures. HAIR12 is involved in the Notch signaling pathway and regulates nervous system development. RANKAL is expressed in the head and scale, regulating osteoblast differentiation. BMP2B and RUNX2B together promote osteoblast maturation and differentiation. The combination of the downregulation of these important genes, LECT1, AIR12, RANKAL, BMP2B, and RUNX2B, is what caused the spinal curvature in zebrafish exposed to lead. Furthermore, researchers found that apoptosis occurs in zebrafish due to endoplasmic reticulum stress caused by lead exposure. RUNX2B is an important gene in the endoplasmic reticulum stress pathway, as well as the proteins PERK and CHOP. PERK and CHOP regulate the RUNX2B gene with osteoblast differentiation and maturation. PERK senses unfolded proteins, and CHOP promotes apoptosis. PERK and CHOP were both heavily upregulated at 48 HPF in lead-exposed zebrafish, inducing apoptosis in muscle cells as revealed by the apoptotic staining. Apoptosis in muscle cells as well as uneven sarcomere formation and underossification all contribute to spinal curvature. The study ends by summarizing that lead exposure causes upregulation of ER stress related to proteins PERK and CHOP and apoptosis in muscle cells, and the downregulation of growth-related genes LECT1, AIR12, RANKAL, BMP2B, and RUNX2B causes decreased ossification and somite formation as well as uneven sarcomere formation. To summarize in my own words, lead interferes with genes and proteins on a cellular level and causes spinal curvature by upregulating apoptosis-related proteins and downregulating growth-related genes. Calcium works to save these fish from the effects of lead by flooding the body with calcium so lead cannot interfere with those systems. That's it for this video. Thank you so much for watching and have a great day.